Hello students, welcome back to GDC. In this video, we are going to discuss 20 important questions from your enzyme topic. So in this 20 question, we're going to help you for your upcoming pharma competitive exam. Let's see what the first question talks about. So first question is Km. So Km is what? So first thing you understand what is Km. So Km stands for Michaelis Menten constant. Okay, Michaelis Menten constant. So what you question is the value of enzyme substrate reaction is dissociation constant, substrate concentration at half velocity of the reaction, concentration of enzyme at maximum velocity of the reaction, substrate concentration at maximum velocity of the reaction. So how you can define Km? So Km is basically nothing but this is your Michaelis Menten constant, and this Km basically what? This is the substrate concentration at half velocity of reaction. This is the substrate concentration at half velocity of the reaction. Next question. Muscular dystrophy disease is diagnosed by serum enzyme. Which serum enzyme basically diagnoses the muscular dystrophy? Your option is amylase, acid phosphatase, alkaline phosphatase, yeah, aldolase. Yes, muscular dystrophy. Quickly give the correct answer. Muscular dystrophy disease is diagnosed by which serum enzyme? Amylase, acid phosphatase, alkaline phosphatase, yeah, aldolase. Tell me quickly. The correct answer is yes, aldolase. Okay. So muscular dystrophy disease is diagnosed by which enzyme? Aldolase. Next question. Coenzymes are coenzymes are having the same quaternary structure like the enzyme, structural analog of the enzyme, soluble colloidal protein molecules, या heat stable non-protein organic molecule. Yes, tell me which one is the correct answer. Comment down your answer in the comment section below so that I could check how many of you have given the correct answer. Coenzymes are what? Having a same quaternary structure like enzymes, structural analog of enzyme, soluble colloidal protein molecules, या heat stable non-protein organic molecule. What is basically a coenzyme? Okay, so the correct answer is option D. Coenzymes are what? They are heat stable, non-protein organic molecule. Next question. In enzyme reaction, Km value is also known as Michaelis constant, isoelectric point, velocity of the reaction, decay rate constant. Just now I have explained you what is Km. You comment down the answer below. What will be the answer for question number 4? What is Km value? Km stands for. In enzyme reaction, the Km value is also known as Michaelis constant, isoelectric point, velocity of the reaction, या decay rate constant. So in enzyme catalyst reaction, Km stands for Michaelis constant, Michaelis Menten constant or Michaelis constant. Next question. With what action so in case of large concentration of the product? So what is there? For suppose during the enzyme catalyst reaction when product concentration increases. I have told you in the properties of the enzyme when product concentration increases, what will happen? Inhibition of the reaction, fast of the reaction, slow of the reaction, and non-effect of the reaction. What product concentration increases? When product concentration increases, what will happen? It will lead to inhibition of the rate of reaction. It will lead to inhibition of rate of reaction. Option A is the correct answer. Okay. When product concentration increases, the rate of reaction will be inhibited. Next question. Enzyme which is used in the treatment of leukemia. Enzyme which is used in the treatment of leukemia is urokinase, streptokinase, asparaginase, or pepsin. Yes. Give me the correct answer. Enzyme which is used in the treatment of leukemia. Leukemia means cancer. Okay. So first thing is streptokinase and urokinase. These are called as fibrinolytics. Okay. Pepsin is used as anti-inflammatory. Okay. But this asparaginase basically used as is a anti-cancer agent. Okay. Used as anti-cancer agent. Next question. Which of the following increases the speed of biochemical reaction? Okay. Increases the speed of biochemical reaction. Your options are antibiotics, hormones, enzymes, or monoclonal antibodies. Very easy question. Which biomolecule increases the speed of biochemical reaction? Which biomolecule increases the speed of biochemical reaction? Very easy question. Enzyme, na? Initially, we are studying na enzymes are the compound which increases the rate of reaction. Option C is the correct answer. Okay. Enzyme basically increases your rate of reaction. Next question. An on-competitive inhibitor binds to enzyme substrate complex, only enzyme, only product, yeah, substrate. Yes, enzyme substrate complex binds to, sorry, on-competitive inhibitor binds to enzyme substrate complex, enzyme, product, yeah, substrate. So first you remember what is the difference between competitive inhibitor. So these competitive inhibitor and non-competitive inhibitor both are comes under your reversible inhibitor class. Reversible inhibitor class. What happens when competitive inhibitors are basically binds to the active site where it compete with the substrate molecule for the binding. But if we talk about non-competitive inhibitor, so they are binds to your ES complex. They are binds to what? They are binds to your ES complex. Okay, option A is the correct answer. Next question. The serum enzyme that used to evaluate pancreatic function. So how you can detect your pancreas is functioning well or not? By checking alkaline phosphatase, amylase, aspartate transaminase, or lactate dehydrogenase. Yes, who will give the answer? The serum enzyme which is used to evaluate the pancreatic functions. Your options are alkaline phosphatase, amylase, aspartate transaminase, yeah, lactate dehydrogenase. Let's see. So lactate dehydrogenase, this is for the your myocardial infarction. This is the enzyme for your myocardial infarction. Aspartate transaminase or AST and ALP. This one and this one. This is for your liver enzymes for detection of liver disease. But this amylase, so amylase is for the pancreatic function. So we call it as a pancreatic amylase. Next question. Which of the following enzyme has fibrinolytic activity? Fibrinolytic. Just now we have L-asparaginase. This is anti-cancer. Pepsin, it is having anti-inflammatory. But this urokinase or if option streptokinase is there, then all these are comes under your fibrinolytics. Okay, I hope you guys are understanding what we are discussing. So these are the questions from the application part of the enzyme. Okay, next question. Uh, Enzymes are best defined as organic molecule that inhibit reaction, non-protein catalyst, biocatalyst synthesized by living cell, yeah, inorganic catalyst. Yes, how you can define a enzyme? Enzymes are best defined as organic molecule that inhibit reaction, non-protein catalyst, biocatalyst synthesized by living cell, yeah, inorganic catalyst. Enzymes are Enzymes are best defined as organic molecule that inhibit reaction, non-protein catalyst, biocatalyst, and inorganic. So enzymes are the correct answer is option C. These are the biocatalysts synthesized by living cell. Option C is correct. Next question. International Enzyme Commission classify enzyme into how many class? 3 class, 6 class, 4 class, 10 class. I want to give one task. All of you give all the classes of name according to Enzyme Commission in your comment section. Everyone, those who are watching this video are requested to write all the enzyme names in the comment section. I will check how many of you have given the correct answer. So according to the Enzyme Commission, so we have 6 class. Okay. So recently one class was modified. So here we can 6, but you just remember one more class they have added to the list. So currently they are 7 classes. Okay, next question. Enzymes are primarily composed of carbohydrate, lipids, proteins, and nucleic acid. Yes, enzymes are primarily composed of carbohydrate, lipid, protein, nucleic acid. Enzymes are primarily composed of protein. I told you now all the enzymes are proteinous in nature. All the enzymes are protein in nature. Next question. The term biocatalyst in context of enzymes refers to. The term biocatalyst in context of enzyme refers to inorganic catalyst that accelerate reaction, organic molecule that decreases the reaction, biological molecule that increases the rate of biochemical reaction, or synthetic chemicals that functions as a catalyst. What is the term biocatalyst means? The term biocatalyst means, yes, who can give the correct answer? Comment down your answer. I want each and every questions to be answered by you guys in the comment section. I will check. I always check once this video uploaded. Okay, the term biocatalyst means biocatalyst or also called as, we call it as a enzymes. Enzymes are biocatalyst, na? The term enzyme or biocatalyst means these are the biological molecule that increases the rate of biochemical reaction. Next question. Human body optimum temperature for enzymatic activity is 37 degree, 25 degree, 20 degree, 15 degree. Very easy question. This is primary school level question. All of you have given this answer. Human body temperature should be what? So that its enzyme activity could be function. What is the normal body temperature? 37 degree, na? So 37 degree is a temperature at which our body's enzyme system act properly. Next question. An uncatalyzed reaction involves high activation energy, low activation energy, balanced activation energy, all of this. Yes. Answer this question. A reaction is uncatalyzed. Okay. You can focus on the term uncatalyzed. See, let me explain you this question. When enzymes are given, they basically decreases the activation energy and increases rate of reaction. So enzymes are decreases the activation energy and increases rate of reaction. When a reaction is uncatalyzed, there will be a decrease in activation energy. So here activation energy can be reversed. That means your high energy activation energy will be high. Option A will be the correct answer. Next question. The pH optima of most of the enzyme is between 2 to 4. The pH optima of most of the enzyme between 2 to 4, between 5 to 9, between 8 to 12, yeah, above 12. What is the optimum pH? The optimum pH, yes. Optimum pH for the most of the enzymes are between 5 to 9. Okay, it will be between 5 to 9. Next question. Which of the followings affects the enzyme activity? Which of the followings will affect the enzyme activity? Concentration, pH, temperature, yeah, all of these. Very, very important question. All of them. All the factors. Your concentration of the substrate, concentration of the product, concentration of the then pH of the medium, and temperature. All these factors will affect the enzyme activity. All these factors will affect the enzyme activity. Next question. The induced fit model of the enzyme action was proposed by Fischer, Cosland, Michael, या Gregor Mendel. Induced fit model. Induced fit model. The induced fit model was proposed by Fischer, Cosland, Michael, या Gregor Mendel. The correct answer is Cosland. So Cosland proposed the induced fit model. Okay. Last question. All enzymes are proteinous in nature except ribozymes, transaminase, transcarboxylase, or acyl transferase. All enzymes are protein in nature except ribozyme, transaminase, transcarboxylase, or acyl transferase. Yes, transaminase is a enzyme that is proteinous nature. Transcarboxylase and acyl transferase enzymes are proteinous nature. But this ribozyme is a non-protein enzyme. Please note it down. It is a non-protein enzymes. Option A. Okay. So in this video, we have completed discussion of 20 important questions from your enzyme part. So I hope you guys are understood what we have discussed today. So thank you for watching this video. Stay connected with GDC for more such informative contents. Thank you.